On. Hello, repeat watchers. Okay, so I think everyone is up and running. We've got like loads of people in here. All good. Okay, so this is my attempt at all the AS biology answers, topics 1 to 4, components 2 to 4 if you're OCR, in an hour. Obviously, I've got to have to select the content. Doing a whole year's worth of work in 1 hour is never going to happen anyway, so I had to do some selection process. I've selected everything that's relevant to all the exam boards, same level of detail. There is heaps and heaps in here. I would get your pens out because I would try scribbling down stuff, some notes and bits and pieces as we go along. You're probably going to find I'm moving a bit too quickly for that. I have clicked record. It is being recorded, so we will, yeah, it will be uploaded ASAP. As soon as this one's done, I'll chuck it up there. So hopefully by the end of tonight, if not, then tomorrow morning on the webinars page that you book, it will, instead of where this was to register, obviously it's in the past. So you just click on that button, it will say watch recording or something like that, and it will be there. We'll also email it out to you, I think, in 24 hours, but that's an automatic setting. Okay, so order of the day. What are we doing today? Okay, so common topics: biological molecules, carbs, proteins, lipids, and nucleotides. These are common and come up all the time. So let me just shut my windows because maybe that's not. Just help with a bit of sound. Okay. Transport across membranes, again, super recurring themes. Protein synthesis, DNA replication, pretty important. Cell structure and organelles, probably not quite as major as topics, but I'm going to go through it if we've got time. Mammalian heart or heart and blood vessels, these come up once in a while. Gas exchange principles, pretty damn common. Mitosis and classification. There's a bit of biodiversity that you'll do as well during this, but we'll see how much we get through. I'm going to go as quickly as I can. I'm going to offer you the choice at some stages whether you want to do more questions and slow down a little bit, or whether you want me just to rip through, and then we'll do some questions at the end. Okay, so feedback from last session was that you really liked the extra sort of exam questions and giving you some more time, a bit more interactive. So I've tried to do as much of that as possible. Obviously, that will be slower, and trying to get anywhere near doing a year's worth of content in 1 hour, we'll see how we go. So we're going to recap and summarize the core content. We're going to do super condensed summaries on the topics, and we'll look at lots of past paper questions so you actually get to see it in practice. And I have already started the recording. I'm just going to double check because I'm getting paranoid. Yeah, okay, sweet, perfect. So moving swiftly on. Carbohydrates, in with a bing and a bang. Okay, so that I'm not going to give you too much time to think about this. I want you in your heads to be testing yourself. Some of you may have heard of the testing effect. Trying to figure this out, looking this up in a book, it's going to do you no good. If you don't know, it's okay to not know some of the answers. But let's start with the two diagrams, alpha and beta glucose. I've just labeled A, B, C, D. Don't forget the oxygen here, and we've got the extra carbon atom that goes up here. So I want you to think to yourselves, alpha glucose, either A and C is the same, or A and D is the same, or B and C are the same, or B and D are the same. So I want your pairings, which A and which one. You can leave it in the questions over here. Which two for alpha glucose are the same? Okay, Beth, I've got B and B. Spot on. Okay, so these guys for alpha glucose are both OH, OH, and then these are. I have a really weird way of remembering this. I think of Santa, ho, ho, ho. So the OHs are next to each other. They're both on the bottom, and also Santa comes down the chimney. So with Santa, this is, I write my Santa like this. My alphas instead of A's in Santa, and Santa coming down the chimney, and that's how I remember they're both there. Really weird. It's quite long-winded. And okay, so for beta glucose, let's move on. Okay, so what's different about beta glucose? Basically, you only need to remember that this end changes. So that tells us immediately that E is H and F is OH, and then we actually flip these upside down. So beta is just the other way around. Okay, so this one will be OH and this one H. They don't ask you to draw it very often, but being able to recognize it and link it to generally is going to be cellulose versus starch, which is the question we're going to look at in a minute. Okay, monosaccharides and disaccharides. These are monos. These are monos. They can ask you straight up factual recall. Glucose, they are all alpha, but you don't really ever get to check that. Glucose plus something equals a disaccharide. I'm going to give you the first monosaccharide. as being glucose plus glucose. This is the most common, usually done as a rework around from starch, using amylase into using something, and then it breaks it down to all glucose. So this disaccharide, we've got lots of guesses. We've got fructose, we've got, okay, I can see the right answer. The correct answer is here, maltose. Now, if you're going to remember any of these, number one is the most important. Glucose and glucose is maltose, because they often, maltoses plus maltoses, lots of disaccharides, you make a polysaccharide, and that is starch. And they often ask the question going in this direction: starch with amylase into maltose, maltose with maltase into glucose. So that is the most common. The other two that you need to know, I'll give you the disaccharides. They are lactose. In fact, I'm going to be consistent with my videos. I put lactose on the bottom. And this one is therefore sucrose. If you can think of the enzyme, maltase is an enzyme, sucrase is an enzyme, although we don't really actually call it sucrase, and lactase is an enzyme. That means it must be a disaccharide, because it needs an enzyme to break it down into its monosaccharide constituents. Okay, so I'd say lactose is the easiest here, because it's got lactose in it. It's not lactose though, it's galactose. I've spelt that totally wrong, haven't I? It's just got galactose. So it's got lactose in it. And then the last one, the missing one, I'll offer some questions. Fructose, you bet it is. Everybody jumping in with fructose. It was only the last option though. Okay, sweet. So I'd say knowing these quite important. Okay, so I put up here the bond between all carbohydrates molecules is bing. This one should be like fresh front and center. Carbohydrate and carbohydrate. It could be carbohydrates you've never heard of before, like random sugar, random sugar. How do we know they're a sugar? They've got an ose on the end. Oh, I don't know what I've done there. I've zoomed out. Okay, so bond between all carbohydrates, whether you've heard of them or not, is glycosidic bonds. You bet it is. Okay, so glycosidic. Again, I'm not going to write that in here. If you're watching the repeat, then scribble it down. Pause it, you can stop it in the repeat, and you write these things down. Polymers of alpha glucose. Now, again, if you can try and get these as quickly as possible, what are the polymers of alpha glucose? We've got starch. Okay, we've got starch and glycogen. Can you give me the two compounds that make up starch? Glycogen, yes. Starch, yes. Starch is made of amylose, phosana, absolutely, and one more. Amylopectin is the other one. Spot on, good stuff. And beta glucose. There's basically only one that you guys need to know about, and it is straight chain, microfibrils, very strong, cellulose. You got it. Okay, so where's my question on this? Do I go? I think, okay, I'm doing this now. Polymers of carbohydrate, which is exactly following on from that. I'm going to change color. We've got the carbohydrate down here, and you may have seen this in the videos. We've got their features coming across. So the function of cellulose is structure. Oh, you can read my writing, or support, or strength. Let's do them across. Start. Oh, okay, so we've just gone through the two. Test yourself again. If you didn't know these two compounds, they both begin with A. Test yourself. Try and think of it before I write them in. I'm going to do them in this particular order. Amylose, that's why the enzyme is called amylase, and amylopectin. Okay, what do these do? Well, these are glucose storage in, or for respiration in plants. So glucose storage in plants, and then it's the same glucose storage in animals. Sweet. Okay. Glucose type, or I've just been through this. They're all alpha, except cellulose, which is beta. Bonds. Straight up, they are all going to have the same bonds in it. I'm actually going to put it in red. Glycosidic. Now, in your notes, I would recommend writing it out a few times, because just cement it in your head. I might take a few shortcuts today, because we're 10 minutes in and we've got heaps to do. Okay, these are the facts they ask about these molecules all the time. Basically, they don't give a toss about any of the other features of these things apart from the ones I put in this table. Cellulose, is it branched or unbranched? It is unbranched. And is it straight? Yes, it is straight. They might like you to relate these to their function. I'm going to come back to that in a moment. Amylose is unbranched, but it is coiled, tightly coiled, takes up less space, so you store more, it's more dense store of glucose, you store more glucose in a small area. Amylopectin is branched, and it is, they never ask about that, so I'm not going to put that in. And glycogen, I'm going to put is very branched, more branched. Is it straight chain? Again, they don't ask about that, so I'm not going to put it in. Is it soluble? Well, it is. Cellulose is insoluble, starch is insoluble, and that applies actually to all of these. In fact, I'm going to put a cross because it's insoluble. I put here why. These are all insoluble for the same reason. Can anyone tell me why this is insoluble? This is asked lots and lots. Boom, Adam. Okay, doesn't affect osmosis. Both of you basically doesn't affect the water potential. I would get water potential and osmosis into your answer where possible. If you just store glucose as glucose, great. It's rapidly available. It takes up a lot of space is one downside, but major downside, it's soluble. That will massively decrease the water potential, and water will come in by osmosis. Plant cells have a cell wall, so they might not explode, but an animal cell, glycogen, if we stored glucose, all our glucose as glucose, our cells would explode. Not what you want. So no effect on, do we know this sign? That's a terrible version of that sign. Water potential is what that sign means if you're watching and you don't know. And therefore osmosis. Okay, structure. Let's do a line drawing tool. So we have beta glucose in fact, let's draw it very quickly, not in a line. So this is my glucose molecule. I'm then going to draw another one here. Okay, so we've got one, and then we have the alternate one, and this would obviously continue. If you want me to draw one more, I will. Okay, so we have alternating oxygens on the top, oxygen on the bottom, oxygen on the top, oxygen on the bottom, and that means we get a nice straight line. So if we were to be representing this as a line, we'd get this. And we actually get bundles of these, and they are joined together by what type of bonding, people? How do these things connect to each other? They've got lots of little negatives and little positive charges, and these bonds that link up, these are called hydrogen bonds. Again, pretty commonly asked. What do we call one of these strings? These strings of straight chain beta glucose are called microfibrils. We are coming on to some questions soon, so you best be paying attention. Okay, as I said with amylose, let me draw. I'm going to draw a couple. No, what the hell are you doing, Rich? Don't draw it like that. Okay, so the oxygens are all going to be on the top, and instead of being a nice, in fact, I'm going to just do that again. Draw it flat. Let me get a better idea. Okay, so instead of the next bond being dead straight on, we're actually going to get a kink. It's kind of a hexagon, and then we get another kink. So this bond angle isn't straight, which basically means every time we bond, it's on an angle, and if we keep adding the same angle to itself, we end up with a coil. So this is how amylose is ended up coiled. I've just realized the detail here. Some of you need to know this a little bit more than others, but in these carbon atoms, we consider this one to be 1, number 2, number 3, number 4, number 5 in the corner, and 6 on the stalk. So we call this kind of bonding in amylose 1,4 glycosidic bonding, because it's bonding from carbon number 1 onto carbon number 4. It's exactly the same as it would be in cellulose, 1 and 4, 1 and 4, 1 and 4, only 1 and 4. In amylopectin, we have the same, we have the 1, 4, but we also have a branch, which means we need to jump out the chain somewhere else as well, which means we get 1, 6, which just means if we count number 6, it's this one on the stalk here. So let's say this is number 6, we would get another carbon atom. In fact, that's confusing because that's amylose that I've drawn. Let's redraw it. Again, it's not straight. And so this is my 1, 6 bond. And so we would end up with, it does coil, but you don't really, they always ask about it branching. So it's always going to branch a little bit like this. Glycogen is going to look exactly the same, except it's more branched. Why is it branched? We can relate this to its function. Amylose is coiled because it's compact and we want to get a lot of glucose into a smaller space of time. Amylopectin and glycogen are branched to increase the surface area. So it allows more space for enzymes to get in to break off glucoses faster. So we can hydrolyze, we can break down amylose and amylopectin faster. Well, glycogen is the fastest because it's got the most branch, high surface area. Amylopectin would be the next fastest. Amylose would be the next fastest. Okay, so I think my questions are underneath for here. Okay, so if you want that a little bit bigger, I'll zoom in a wee bit. Describe how the structure of starch and cellulose molecules are related to their functions. Okay, so what have we got? Analyze the question. First of all, command word is describe. We're giving a description, not giving, I think you kind of need to link it. So there's some reasons going in here. Starch, immediately, bing, 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 amylose, amylopectin. They are going to be in your answer. And we're going to link the structure to the function. Okay, so I'm going to give you an insanely short amount of time. I'm going to give you 2 minutes. You can bullet point me. In fact, leave it in, I don't want separate answers. If you can write one continuous text box, maybe separating the commas or slashes, and I will pick out some of the answers and I'll show you the mark scheme in just a second. So again, we're trying to link the structure. Remember those words that have just been in that table. They are not there for, because I made them up, because I think that's what they're going to say. I know that they are the features that they want you to talk about. So what were the column on the left-hand side? "20 past already." Behind my schedule. "30 seconds. A window pane bigger. Too many answers coming in. Okay, it's a minute. So we've got lots of amylopectin is branch, allows faster access for enzymes to hydrolyze and produce ATP. Oh, it's just gone off my screen. I can't finish reading it. Sorry. I'm going to pick one down the bottom. Well, okay, amylose is straight and coiled, which makes it compact. Amylopectin is branch, which increases the surface area for glucose to be used for respiration. I've lost it. It keeps the too many responses. I can't go through them all at once. Okay, back to it. Amylopectin is branched because it's 1,4 and 1,6 glycosidic bonds. Lots of ketones, very nice. This increases the surface area for enzymes to get in, hydrolyze the bonds. results in rapid release of starch, with the same with glycogen, but it's more branched. Okay, do we need to be mentioning glycogen in this answer? We're just looking at starch and cellulose. So we've got to be really careful to try and link this as much as possible. Okay, so let's take a quick look at the mark scheme. Okay, so we can get 5 marks. We can get a maximum of 3 for each. So you can't just pick one and talk about one. Otherwise, you'll get a maximum of 3. So helical or spiral or coiled, so it's compact. It gives you 1 basically for saying compact. This is a nice word that tends to come up quite often. It's a large molecule. It's insoluble, so it's osmotically inactive. So it doesn't affect water potential, except does not affect water potential. Branched, so glucose is easily released for respiration. So it needs this. See here, we've got so in brackets. We need to link. The question told us that we need to describe how the structures are related to their function. So it's looking for that linking in the answer. Large molecule, so it cannot leave the cell or cross the cell surface membrane. I've never seen that one before. You don't have to. Obviously, it's 5, and there's 8 possibilities on here, so we don't have to get them all. Cellulose, long, straight, unbranched chains of beta glucose. They love you to talk about beta glucose when talking about cellulose because it is the only one you really have to know about. Joined by hydrogen bonding to form microfibrils, provide strength or rigidity. So they always want you to link hydrogen bonding to the strength to its function. Okay, super good. Moving over. Okay, so proteins. Let's have a quick look. I'm going to get, okay, so if your name starts A to N, you can think about primary structure and secondary structure. If you're whatever, O to Z, let's have a think about tertiary structure. You can give your answers. I want a single bullet point or sentence description of primary structure, secondary structure, tertiary structure, or quaternary structure, depending on which group you are. I'm going to start with a little diagram. Okay. Abigail, very nice. Okay, primary structure is a sequence of amino acids. It's textbook definition. Alpha alpha is my shorthand for amino acids in the chain. So here the diagram is like this amino acid square, circle, triangle, triangle, square. This is the particular sequence of amino acids. What bonds are involved in that? Has anyone told me that? No, I haven't asked you for that yet. Okay, let's do the description. Secondary structure. Okay, dude has told me. Can't see anyone with secondary. Okay, so we're perfect. We've got regions within this fold up into alpha helix. Technically helices if we're talking in plural, but they don't mark you down for things like that. Beta pleated. You don't have to say pleated, but they normally do. Beta pleated sheets. So that means just that regions, we've got massive long chains of these things, and regions of them, because of their bonding, regions of them coil up, and we call those regions alpha helices, and some regions of them fold back on themselves in like big layers and stacks, and we call those beta pleated sheets. And those individual areas, when we look at them on their own, we call alpha helix or beta pleated sheet. I should probably have done that in green and red, but never mind. Tertiary structure. I've seen some of you with tertiary structure. It's the 3D structure of a protein, held together by, we've got the bonds over there. It was a very nice description, Rima. Okay, so it's, I'd say, the 3D shape of the protein is fine. We'll come to bonds in a moment. And let's pick someone. Tertiary structure is the final 3D resting shape of the protein. Someone's been watching my video. I think that's exactly the words that I used. Quaternary, several different polypeptide chains, Rebecca, such as hemoglobin, giving me an example. Very nice. Okay, so this is more than one polypeptide chain. Absolutely, it is. So hemoglobin, collagen, fibrous and globular proteins, AQA don't do the difference between those, so I'm not going to go into too much detail there. So if we did hemoglobin, we've got 2 alpha, and they interlink and join, but these are made separately, different translation, and then they're joined together. They're packaged up in the Golgi apparatus. Should probably use different colors. Okay, let's go. Bonds. Bonds. What are these bonds here? These bonds between amino acids. Absolutely, everyone should be going bing, 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 bing. Peptide bonds. Awesome from basically all of you. Very common that this comes up. Which bonds cause the alpha helices to form and cause the beta-pleated sheets to form? Okay, Sophie, spot on. Hydrogen bonds. Okay, so I'm going to put in here peptide because the peptide bonds don't cause the... Which bonds are present? Well, this is a chain of amino acids, so peptide bonds are present, but what actually cause the secondary structure are hydrogen bonds. Forgive my handwriting, I'm going really quickly, but I think I'm wearing out the nib of my digital pen. Tertiary, okay, so I'm going to do peptide every time. All the previous ones are going to be included, and hydrogen bonds. Now, different examples are going to give me slightly different answer here, but the ones that are common to all of you, there's probably at least 3. So we've got ionic bonds. Okay, these are like real bonds. They are quite strong. The strongest of them all, anyone know the strongest of them all? Imogen, absolutely, disulfide bridges. Oh, bridge. These form between cysteines, just as a little note. Not mega important, you don't have to know that. They often tell you that cysteine contains sulfur, and this protein has lots of cysteines in it, suggest about its properties. Well, it's probably going to be strong or stable because it's got lots of disulfide bridges, it's got lots of cysteines in it. And there's also there are some covalent bonds in there, but not very many. And there are some hydrophobic and hydrophilic interactions. If you're an AQA student, you don't need to know that specifically, I don't think. So that just means that some regions of it are hydrophobic and they don't like being in water, so they end up on the inside. They just run away from the water, so they fold on the inside, and the bits that are hydrophilic that like being in the aqueous environment of the cell are on the outside. That's all that means. Quaternary, all of the above, same. Okay, where do I go from here? Do I go down? Do I go up? No, my arrow. Oh, okay. So structure of an amino acid, we've got A and B. B is going to be our carboxylic acid group, or carboxyl. R group is obviously variable. It changes depending on the amino acid. That could be a very long, complicated string of things. It could be another hydrogen and glycine, I think, is the one with hydrogen just on the end there. And this gives us the acid bit because it's a carboxylic acid, and this gives us the amine bit. This is the amino group. And why are proteins special? Because they contain, that's not a very good standout color, they contain nitrogen. So all the biological molecules contain carbon, hydrogen, and oxygen in various different proportions. Amino acids and proteins contain nitrogen. Again, very commonly pops up. Okay, where am I going? No, I think I'm going to go across and then up. 6:30. Describe, okay, so this is just going to show you in words directly off a mark scheme. None of this I have typed. I've taken this all off. This is, I think, an OCR paper, but the level of detail you need to know is pretty much the same with the potential caveat of the hydrophobic hydrophilic interactions. Okay, so describe the structure of a hemoglobin molecule. 7 marks. Primary structure: sequence of amino acids joined by peptide bonds. Boom, it's in the table. That's what you need to know because that's what they always ask. Secondary: alpha helixes, small regions of beta pleated sheets are folded up, held together by hydrogen bonds. Okay, that's what you need to know every single time. It's converting that table into short, concise bullet points. Tertiary structure is the secondary structure helix or polypeptide change undergoes further coiling or folding. The bonds or interactions form disulfide bridges, ionic bonds, hydrogen bonds, hydrophobic or hydrophilic interactions. Hydrophilic, the bits that like water are groups, which is obviously the bits on the top, the bits that change between the amino acids. Hydrophilic ones on the outside, the hydrophobic ones on the inside. There's a greater emphasis on OCR spec than the other specs on the hydrophobic, hydrophilic stuff. Quaternary structure, well, this is hemoglobin. It's a quaternary structure. OCR are required to know the specific structure of it. I think EdXL2 in the globular proteins section. AQA, don't worry too much if you don't know the ins and outs of hemoglobin. 4 polypeptide chains, as I drew them, 2 alpha, 2 beta chains, 1 heme group, and it's a prosthetic group again. Prosthetic, don't stress if you're AQA, it's not in your specification. Moving on. Okay, so this is, okay, HIV, I'll make it a bit bigger, and some of you have said that you can't see very well. Amine or amino, the amine group or amino group, I mean, it's called an amino acid. I would call it it's the amine, oh, you're quite right. Did I write amino group? It probably should be the amine group if I'm being technically accurate. Amine group, and it forms an amino acid. Good point. Good little spot there. Okay, let's read the question first. HIV, it's a virus. Okay, so what's going through my head as I read this question? HIV, it's a virus. Attaches to a specific protein receptor on a helper T cell. Okay, so at this point, this question could be about a few things. A low percentage of people have a mutation of the CCR5 gene, which codes for this protein receptor. So this gene codes for the receptor. The mutation results in a non-functional protein receptor. Explain, give reasons how this mutation can result in the production of a non-functional protein. Okay, so now we're going to do an exam technique webinar. What's this question on? Is this question on HIV and immunology? Is this about the immune system, or is this question about mutations and proteins? This question is about non-functional proteins. It's got basically nothing to do with HIV. You don't need to mention HIV in the question at all. Okay, so how does a mutation cause a protein to be non-functional? This could be a receptor. It could be an antibody. It could be an enzyme. And protein shape is essential to their function, and it is important that whatever the protein does, it has its specific tertiary structure. And therefore, they can ask us about any type of protein. Okay, so we're looking 4 marks. We're looking for at least 4 bullet points or 4 sentences. In my past papers, I always do bullet points. In the real exam, I write short, full sentences, and I just use a full stop like a bullet point. So I might finish my sentence here, and then I'll start my next sentence just like a short bullet point. But I obviously don't write in bullet points. It doesn't look that great. Okay, so these questions should become more and more second nature the more practice you do. So mutation. Change in the sequence of DNA, change to the sequence of messenger RNA, change to the sequence, the primary structure of amino acids, change to the tertiary, different internal bonding, change to the tertiary structure, no longer complementary to the, whatever it is a receptor, isn't it? Okay. Change in DNA or nucleotide sequence, change to amino acid or primary structure of the protein, change in the position of different bonding, change in the tertiary structure. Every time. Same model answer, all exam boards. Comments are sometimes useful, except mutation in DNA base sequence, except deletion, substitution, addition. There is a bit greater emphasis on the types of mutation in the new specs for all exam boards. Reject different amino acid formed. It's looking for the sequence of amino acids, and it's looking for the effect of that on how it affects the bonding. Reject any reference to active site because this is not an enzyme, this is a receptor. It says ignore 3', so just don't, you can use this in your notes, I use shorthands all the time, but in your exam, write tertiary. So why are proteins specific? This could be enzymes, antibodies, receptors. It's the same answer, basically. These are, again, off a mark scheme. Glucose biosensor detects only glucose. Use your knowledge in the way in which enzymes work to explain why. So this question is just saying, why are enzymes specific? The enzyme active site has a specific tertiary structure. Only glucose or the substrate has the correct or complementary shape to the active site, for another mark, forming an enzyme substrate complex. Let's have a look at another real past paper question. Tests using monoclonal antibodies are specific. Use your knowledge of protein structure to explain why. Look at how similar these questions are. Specific primary structure, order of amino acids. Specific tertiary structure, or 3D structure, only binds to fits to the complementary to one antigen, forming an antigen-antibody complex. Same answer, same thing, different, slightly different key terms. Good. So I need to ask you some more questions. I've been kind of ignoring you. Okay, so we've still got heaps and heaps to do. So it's why I'm trying to go. Okay, so this is what I want. I want you to name A, B, C, D, and E for me. I'll zoom in a wee bit. Okay, so A, what is A? Mystery molecule. B is the bond. C, be specific with C and D for me, please. There's a difference between them. There's a generic one which we could write, we could label as in the middle, this middle block here. I've just done a block because I couldn't be bothered to draw them, and I want to know what this entire is, the entire molecule. Okay, Rachel, A is glycerol, correct. Not a phosphate group. There is a difference that we're going to look at, the difference between triglycerides and phospholipids over here. B is an ester bond. Think in fats, carbohydrates have got glycosidic bonds. Proteins have peptide bonds. Fats or lipids or triglycerides have ester bonds. Think of a fat ester woman wearing polyester, big fat sweaty woman. C is a saturated hydrocarbon fatty acid. Spot on. I should give some names, shouldn't I? E, Ariba, you are quite right. E, the whole thing is a triglyceride. And let's pick someone at Yama. C is saturated fatty acid. What about D? I've said Rebecca lots. Duda, are you spot on? Yeah, unsaturated fatty acid. Okay, it's unsaturated because it's not completely full of hydrogen. We could break this hydrogen bond. Each carbon can make 4 bonds. Okay, so 1, 2, 3, 4. I'm going to remove them because they look like double bonds now. But each one can make 4 bonds. So 1, 2, 3, 4. These carbons are currently occupied. If we were to break this bond, we could add theoretically more hydrogen. I've missed a hydrogen off there. Okay, so that's why we call it unsaturated. It's not totally full. I haven't drawn a phospholipid. This table asks us for, this is an OCR exam. Again, you will need to know this kind of stuff. 2 differences and 2 similarities between a triglyceride and a phospholipid. I haven't been asking you many questions, so I'll give you 1 minute. Reset, go. So difference. This difference is going to be the same. So if you decide to write, I'm not going to say anything. I'm going to let you think. Very quick, some of you. Liberty. Spot on, Liberty. Sophie. It's hard to read moving text. Exactly the same as Liberty, spot on. Same for you, Deborah. Okay, right. 1 minute, spot on. Okay, so there's there's not lots. There's a few things that we could say. Start with the things you know. Sometimes these are going to be quite difficult to sort of put into words. If you need to just do a little sketch over here, it's got glycerol, fatty acid, phosphate, Oh, that's not very good. Glycerol, glycerol, fatty acid, fatty acid, fatty acid. Okay, so what have we got? Well, we've got 2 fatty acid tails, 3 fatty acid tails. So there's a difference. Oh, wrong way around. 3 and 2. We'll see the answers underneath. Well, what's this? This is, it's like spot the difference. It's easier to do when you do it out. So no phosphate group. It's PO4 3 minus. It's not 3 plus. It's 3 minus. And then does have. And you can't call it phosphorus. You call it the phosphate group. They're quite hot on that. They don't like you calling it phosphate because that's an element. Phosphate is just P. This is a phosphate group. It's PO4 3 minus. It's a difference. Similarity. Well, we've got glycerol. Both contain glycerol bonded onto fatty acids, but they both contain ester bonds. You could say triglyceride has 3 ester bonds. A phospholipid only has 2 ester bonds. Let's have a look at the mark scheme because it will give you. Okay, let's finish the question first. I'll come back to that in a second. Name the test used to identify presence of lipids against single 1-point factorial recall. It is the emulsion test. The ethanol test. I would call it the emulsion test. I'll look at the mark scheme. I'm pretty sure I know it uses ethanol. I know that you understand what the test is, but I would think the emulsion test is the name that they want. 3 marks. Describe how you would carry it out. 3 sentences. All of this, wasted space. You do not need it. This should be, if you're writing more than 3 lines for this, then you don't know the answer concisely enough. Yeah, add ethanol, mix, shake, stir, pour to water. The emulsion test for Edexcel, Imogen, yes, you do. You have to know the test for the biological molecules. I will double check, but let me just get rid of this. I can find that right now. I'd be amazed if you don't. AT spec. Oh. Oh, Oh, I'll find it and I'll let you know. Crush food sample. Okay, so if they say you've got a seed, how would you tell if the seed has this thing in it? Then you crush the seed first because otherwise it's gonna to increase its surface area, release all the goodness. Yeah, okay, really nice. We're gonna check the mark scheme in just a minute. What would be the result? Milky emulsion. Yeah, so a milky or cloudy will be the answer here. Where is the answer? Okay, so this is the mark scheme. The differences, the total differences: 3 fatty acids, 2 fatty acids, 3 ester bonds or 2 ester bonds, absence of phosphate, presence of phosphate. Similarities: contain glycerol, contain fatty acids, contain ester bonds, contain the elements carbon, hydrogen, and oxygen. What is the test called? Emulsion test. Does it allow alcohol test? Does not allow alcohol test. There is another test called the Sudan 3 test, but no one ever teaches it anymore. The emulsion test: add ethanol, shake, stir, or mix, add to water. You will see a milky or cloudy layer. Wow, we're at 6:45. I need to get. Okay, so I'm going to give you a choice now. I can keep asking you questions, and we can probably not get through everything, or I can like rinse through and give you like more of a lecture, but it will be less. Just whiz through, says Farzana. Rinse it, no questions, go through everything. Okay, unanimous decision. Okay, so I'm going to just basically do all the answers for you and just give you a summary. Structure of DNA, we've got a phosphate group. Again, phosphate, not phosphorus, it's PO43 minus. We've got the sugar in DNA is deoxyribose. Okay, it's a sugar, it ends in OSE, it's a pentagon, it's a pento sugar, but if they ever ask if it's DNA, they want to know the specific sugar. This is one of 4 nitrogenous bases. We have adenine, we have thymine, we have cytosine, and we have guanine. You should be pretty familiar with these by now. If a DNA, the quite common question is 12% adenine, what percentage of it is guanine. How do we work this out? Well, we know that for every A, there's a T. So that means that there's 12% A, there must be 12% T. That means there's 24% in total A and T. So we do 100 minus 24 gives me 76. And we now know that, does that work? Have I done my maths right there? Probably not going, yeah, 78. Good maths doing this. No, 78. This is 24. 12, 24. Maybe my numbers don't work. I just made this up off the top of my head. Let's make this 18%. So that means this is going to be 18%, which means this is going to be 36%, which means we're going to have 64% is going to be C and G combined. So we divide it by 2 and end up with 32% G or C. That's how you do those. They all come up quite nicely. Okay, name the bonds in a polynucleotide. So these are going to form DNA or RNA. The bonds between, if we were going to make another one down here and put more bases on, we form the sugar phosphate backbone. And we form these bonds that here, and they're called phosphodiester bonds. Okay, so the bonds always come up. The bases, we've got adenine, we've got cytosine and guanine, but we have uracil instead of thymine in RNA. Okay, so that is, did I put any questions down there? No, I didn't. Okay, so transport across membranes, diffusion, they always want basically a couple of bullet points only. So what type of molecules? They always want to know what kind of molecules, they always want to know which directions. So this is in and out of a cell. So diffusion happens anywhere in air from high to low concentration, we know that, but across a cell membrane it's going to be small, non-polar. So, or fat soluble, lipid soluble, because it's made of a phospholipid bilayer, so anything can dissolve through, lipids can go through, and it's obviously from high to low concentration. Osmosis. Okay, ask yourself the question, the red is a solute such as glucose, the blue is water. Which way does the water go? Very, very commonly mistaken. So it goes, what type of molecule? Osmosis. Water is the molecule is moving. Which direction? From high water potential to low water potential. But which one is low water potential? This one is the low water potential, so this one is high, so it's going to go in this direction. It looks counterintuitive because you've got more solute here. It looks like if the solute could move across this membrane, the solute would the solute would move across, but it can't. Water's the only thing that can move, so it tries to dilute this one because it's super strong. Orange arrow, correct, Adam. Okay. Facilitated diffusion. Okay, so there are two types. I want to know the name of this protein, and how they work. Okay, so small charged particles. So what type of molecule? Small charged. So ions, basically. Oh, terrible handwriting. Yeah, and they go through a channel protein. Yeah, it's a narrow channel. It goes through the channel. Now we've got large molecules, basically glucose, and they're going to go through. Think of an aircraft carrier is a massive boat, so these are carrier proteins. And they bind to its specific shape, go through all the rest of it, normal protein style questions there. Active transport is exactly the same as facilitated diffusion with a carrier protein, except it's against the concentration gradient using the ATP. Whenever you see active transport being mentioned, you want to say it's against the concentration gradient. That's conc grad for concentration gradient. I can zoom in maybe just a little bit more. And then it's using ATP. Which type of protein is it? It's a carrier protein. It's exactly the same as facilitated diffusion, except it's against the concentration gradient using ATP. So let's have a look at this question. I'll rip through this. I'll put red as my answer. Some molecules move across cell surface membranes by passing their concentration gradient through the phospholipid bilayer. The movement of some polar molecules across the membrane involves carrier and channel protein molecules. When this movement occurs down a concentration gradient, this is called facilitated diffusion. And when it occurs against the concentration, the process is called active transport. Energy from in the form of ATP is used in the movement of molecules against concentration gradient. Active transport against the concentration gradient, ATP, all the time. It's a carrier protein. Gas exchange. How are organisms adapted for efficient gas exchange? There's three major factors. It doesn't matter which order you put them in, but surface area, whopping great one. We have diffusion distance, or length of diffusion pathway, if you prefer to phrase it that way. And some people often say temperature. Temperature increases the speed of diffusion, yes, but in both directions. The last one is concentration gradient. Whoa. Let's just undo that. That is horrendous. Okay, so how are biological organisms adapted to maximize these things? Okay, so surface area. We've got the alveoli of the lung, increased surface area. We've got the gill filaments and lamellae in fish gills. We've got the branching of the trachea in insects. We've got villi in terms of internal digestion. Absolutely, I was thinking of gas exchange, but villi or for internal digestion. Flatworms to increase the surface area for their respiration, to dissolve oxygen across their surface. Okay, so diffusion distance, we've got thin cell membranes. So alveolus epithelium, 1 cell thick. Capillaries are their linings are 1 cell thick because they want to decrease the diffusion distance. So these are all the adaptations they want to give you. They want you to list these adaptations, and they want you to link anything that's basically exchange related. In fact, you can almost remove gas from this. These are the principles behind every question, and then they're often looking for you to link it to one of these. Concentration gradient, this is going to be ventilation, which means moving the air generally around, and circulation, moving the liquid around, which is generally going to be blood. And obviously that removes the air that's, let's say, low in oxygen and replaces it with air that's high in oxygen to maintain a steep concentration gradient. And the blood circulation is going to remove the blood that's high in oxygen and replace it with blood that's low in oxygen. So again, you maintain a steep concentration gradient. Describe the process of. Now these are actually, well, transcription and DNA replication have a lot in common, and translation comes up much less often. Transcription way more common as a question, DNA replication pretty often as a question. I'm just going to scoot straight through these. I'm going to just read through these answers, so they're going to show you how similar. There's OCR, Edexcel, we've got an AQA version of DNA replication over here. Describe how a nucleotide base sequence in a gene is used to synthesize a polypeptide. Okay, so this is a manky question, but describe how the base sequence of DNA is converted into making a protein or a polypeptide. So you've got to know these key terms in order to understand the question, but let's just go through it. Transcription. DNA of gene is copied or transcribed into messenger RNA. Key terms, key terms, key terms. Free or activated RNA nucleotides. So free RNA nucleotides are added according to complementary or specific base pairing. So the template strand is going to be the one on the DNA, and the RNA nucleotides will be added one at a time based on which one they're pairing up with. Might be worth mentioning that A pairs with U in RNA. One strand is the template strand. Can also call it the sense strand, and it's catalyzed. The enzyme is RNA polymerase, and it's forming, again, phosphodiester bonds. Doesn't hurt to have too many key terms in there as long as they're relevant. Translation. Okay, so 7 total. You can get a maximum of 5 for transcription. Messenger RNA, I would say, moves into the cytoplasm because it leaves the nucleus, moves to a ribosome in the cytoplasm. Transfer RNA binds to the messenger RNA. The anticodon on the transfer RNA is complementary or matches or pairs to the codons on the messenger RNA. Common mistake, people refer to codons in DNA. Codons don't exist in DNA. They're triplets, but codons are messenger RNA. So the codon matches with the anticodon, and the tRNA has a specific amino acid attached. And then again, what do you always talk about when making a protein? Peptide bonds form between the amino acids. How do Excel phrase it? Basically exactly the same. Describe the sequence of bases in DNA molecule that would be used to form the primary structure of a protein. So describe how the sequence of bases in DNA would be used to form the primary structure of the protein. Same question, different wording. Sequence of bases forming the genetic code determines the amino acid sequence. The idea of one triplet code codes for an amino acid. DNA acting as a template strand, reference to transcription or transcription of DNA, e.g. DNA unzips. The idea that messenger RNA moves from the nucleus into the cytoplasm, reference to translation. So again, if it doesn't have transcription and translation in the question, definitely put those in your answer. The role of a ribosome, codons or anticodons. Transfer RNA carries the amino acid. The formation of peptide bonds between amino acids. The primary structure is the sequence or order of amino acids. Okay, so really, really similar. And how is it similar to DNA replication? Well, let's look. The strand is separated, the two strands separated, or hydrogen bonds break. This is done by DNA helicase. They haven't actually mentioned DNA helicase, but it's the same enzyme to unwrap the DNA, unzip the DNA. Both strands act as the template strand. So that's a difference between transcription and DNA synthesis or DNA replication. Free nucleotides attach, again, according to complementary or specific base pairing. Should always be mentioning this when you've got A's and T's and G's and C's joining up. DNA polymerase joins nucleotides, so this time it's DNA polymerase. Again, we're forming phosphodiester bonds. Hydrogen bonds reform, that's kind of a minor point, to be honest. Semi-conservative replication, whenever you're talking about DNA replication, semi-conservative, because each new molecule contains one old strand and one new strand. There's a diagram from a past paper. It says this is a ribosome, this is messenger RNA, this is transfer RNA, and this is an amino acid. Tetracycline is a molecule that obviously binds to the messenger RNA and sits here. Tetracycline is an antibiotic. The diagram shows how tetracycline binds to bacterial ribosomes. Protein synthesis in bacteria is similar. How does tetracycline stop protein synthesis? Do you need to mention DNA ligase, Amy? DNA ligase is most commonly used in the gene technology section. DNA ligase is used on the antisense strand of DNA to rejoin up the small sequences. I will potentially, I don't think they're going to ask you about DNA ligase, which is why I didn't put that section in. So they can. DNA ligase is the enzyme that joins sections of polynucleotides together. It rejoins up double-stranded DNA. I'll come back to that. Amy, if you're still worried about that at the end, let me know. What is the difference between a nucleic acid and... I'm going to come back to questions at the end. I think it'll be way quicker. So tetracycline is an antibiotic. How does it stop protein synthesis in a bacteria is the question. Tetracycline binds to or the messenger RNA codon. The anticodon cannot pair with the codon. The amino acid cannot be added to the polypeptide chain. Translation, again, it's not in the question. Translation prevented. Right, there's loads on protein synthesis. Sense and antisense. Depends on your exam board, which is why I didn't go into too much detail there about sense and antisense, but they can. I'll maybe come back. There's two questions on sense and antisense of DNA ligase. I'll never come back to the end. Mitosis. When during cell cycle does DNA replication take place? In most of the questions when they ask about mitosis, interphase. Not during mitosis. It happens if you need to be specific at the end of S phase, but go with interphase. There's a bit more detail on cell cycle this year, but interphase will be the answer they're looking for, unless it says which phase of interphase does it take place, in which case it's S phase, synthesis. 2 bullet points for prophase. What does prophase? You need to know the characteristic features of these stages. So the nuclear envelope breaks down. Chromosomes condense. or become visible. You may say the centrioles move to the poles. It's a minor point, but it could be a third point. Metaphase, what happens next? The chromosomes line up on the equator, and the spindle forms. You could say attaches to centrioles, centromeres. I nearly made a mistake then. Centromeres. Anaphase, the spindle contracts. I'm going to say chromatids. Pulled to the poles. And you can say the centromere splits. Probably should have put that one first, shouldn't I? But never mind. Can you say nuclear membrane? Yes, you can say nuclear membrane. What's the difference between centrioles and centromeres? I'll draw a quick diagram. It's on the next slide. Telophase, the nuclear membrane. I'll use it membrane this time. Reforms, and the cytoplasm divides, which is called cytokinesis. Cytokinesis. If you can't read my writing, you're going to have to just Google them because you'll figure it out. Okay, so what is the difference between a centromere and a centriole? Okay, so one, two, three, four. What are these things? One is an interesting one. I don't know why I put that one first. This is the position of a gene on a chromosome is called a locus, or loci if you're speaking in the plural, L-O-C-U-S, locus, like location of a gene on a chromosome. Two is the bit that joins the two number threes together. That is called a centromere. And I help remember this. Mere, chromosome, has an M in it, so centromere is on a chromosome. Three is a sister chromatid. It's half of it. These are identical copies of one. This half is exactly the same as the other half because DNA replicates during interphase. And four is the centriole. Centriole moves to the poles. So each end, they look like T-shapes, like this. Okay, so what phase? You need to be able to recognize these from terrible cell images. I think you're right, Ronan. A is metaphase, so metaphase in the middle. You can think of metaphase, if they're lined up down the middle, it is metaphase. B, what's happening here? Well, we've got the nuclear envelope breaking down. We can see the chromosomes. B is prophase. C, super easy, the easiest one to spot. This is the cytokinesis, or the cell splitting. We've got the nuclear envelopes back again. This is telophase. D, the characteristic features of this one is the triangular shape, or the being dragged towards the poles, and this is anaphase. So they are the things to look out for. They ask that relatively often. Let's zoom out and see what's next. Okay. Whoa, not that much. Okay, so I'll just leave this on the screen so you can come back to this. Describe the events that occur during one complete cell cycle, including mitosis, in an animal cell. This is a WJC paper, but same detail. Interphase, replication of DNA. Always want to know that. Also some replication of organelles. Nice to know. Not a massive point. Synthesis of ribosomes, proteins, ATP, getting ready, basically preparation for a busy time ahead, A, B, and C. Prophase, chromosomes condense, joined at a centromere, nuclear membrane disappears, so that's prophase. Chromosomes line up on the equator during metaphase, spindle formation is metaphase, that's what we're looking for there. Centromere divides, splits at anaphase, the chromatids are pulled to opposite poles during anaphase. Again, so that's all done. Contraction or shortening of spindle fibers, so those three are all related to anaphase. Last one, nuclear membrane reforms in telophase, cytokinesis or cell division. So they are what they're looking for basically all the time. Okay, so where are we at on my list of things? ribosomes. Just jump over here. We're nearly done, actually. I've got through quite a lot of content already. Okay, so this question is basically some organelles. This question here, let's zoom in on this one, is basically describe the difference in structure between a bacteria and a eukaryote. So let's look at the question and see how they ask it. The structure of a cholera bacterium is different from the structure of an epithelial cell from the small intestine. Describe how the structure of cholera is different. So they haven't used the words specifically, but this is, how is a bacteria different to an animal cell? It doesn't, you don't really have to go into the specific details about epithelial cells. Can a bacterium have RNA too? They can have RNA, but I would say these are kind of like the model answers. First of all, nucleus. Major difference between a prokaryote and eukaryote. Eukaryote means truly nucleated. They have a nuclear membrane when the DNA is inside it. The prokaryotes have a single loop of DNA or a single circular chromosome, sometimes they say. And that's the one major difference. Probably the first front and center, bang, that's the easy answer. Then you're allowed any two from no membrane-bound organelles, which means all the organelles that inside them that have membranes, so mitochondria, Golgi apparatus, it'd be chloroplast, endoplasmic reticulum, these all have membranes. Little point to point out here, I would mention two of these, so no mitochondria, no Golgi. All cells have mitochondria, all eukaryotes have mitochondria and Golgi. Small ribosomes only in prokaryotes, large ribosomes only in eukaryotes. And then again, you're allowed two from the optional things that sometimes prokaryotes have, so a slime capsule or a capsule, a flagellum, although sperm cells have a flagellum, so I'd steer clear of that one. Plasmids are prokaryotes only, and cell wall, maybe give the material, peptidoglycan for prokaryotes, and cellulose would be for plants, obviously. I won't go into chitin, not all of you do chitin. This one is about organelles, but it's also a little bit about proteins and how proteins are made. So it's kind of linking the organelles that are involved in making proteins in sequence from start to finish. This crops up more often than you would think. Pancreas is an organ that secretes protease enzymes. Okay, protease enzymes. Outline the organelles in the pancreatic cells. The organelles in the pancreatic cells work together to produce and release protein molecules from cells. So what's this saying? This is saying, how does a cell make and get and excrete an extracellular enzyme? Doesn't really matter what the enzyme is. We just need to know how is a enzyme made? What is an enzyme? An enzyme is a protein. What are the organelles are involved in doing that in order? So basically going to be obviously we know with transcription translation, we're going to start with the DNA in the nucleus. We need to mention the organelles. So DNA in the nucleus, we're going to go messenger RNA goes to a ribosome. If you want to mention transcription and translation, you can. I don't think you're going to get any marks for it here. Ribosome, this is probably going to be on the rough endoplasmic reticulum, which is probably going to be attached to the nucleus. And this is going to form the protein, which is then going to be put into a vesicle, which is going to go to the Golgi apparatus or Golgi body, same thing, different terms. It's then going to be modified, is what they normally say here. And then it's going to go into another vesicle, and it's going to go to the cell membrane. It's going to fuse with the cell membrane, probably by exocytosis, I imagine. They might not like exocytosis, but we'll see. And then it will be outside the cell. So if you can basically, obviously you're not going to write bullet point answers like this in the exam, but you are going to, this is the point. This, like, the point of this sentence is DNA in the nucleus, full stop. Once you've got a short sentence, like no more than one line maximum for each of these things. Let's have a look at the mark scheme and see how well I did. Okay, nucleus contains genes, site of transcription produces messenger RNA. Nucleus is underlined, it must be in there because it's looking for the organelle. Ribosome or rough endoplasmic reticulum. RER is the site of protein synthesis or translation. Vesicles for transport. Golgi apparatus modifies or repackages the protein. Vesicles fuse with the cell surface membrane. It says ignore exocytosis. Bad, Richard. And this was actually a quality of written communication, but that's not. I'm assuming you're going to write sentences in a sensible order. So if you write in an incorrect order, if you started this with Golgi and then you went to the endoplasmic reticulum and then you went to the nucleus and then you went to vesicles, like if your answer's all over the shop, that's where you're going to lose those marks for quality of written communication. But if you put it in a sensible order, then you're not going to be doing too badly. Okay, so last couple of topics. This is the heart viewed from the front. What are the blood vessels? A, B, C, D. What are chambers 1, 2, 3, 4? And what are the valves X and Y? Okay, let's start with A, shall we? It's coming back towards the heart, so it must be a vein, which gives us a clue. It begins with a V. This is the vena cava or the vena cava, depending on how you want to spell it. The blood's going to come in here. It's going to come into the left and right reverse. This is from the perspective of the person who owns the heart. We're looking at someone else's heart, so this is actually, it's not the left, this is the right atrium. This is the right ventricle. Blood comes through the atrioventricular valves. If your teacher's gone with bicuspid and tricuspid, fine, they're not wrong, but this is the I never teach them because they're confusing. I think it's tricuspid and bicuspid. You can get them wrong. You are allowed to say atrioventricular valves, the valves between the atria and the ventricle. It's almost impossible to get wrong because the name gives you the clue. So I would say atrioventricular valves all the time. Then blood comes up and out of B, which is going to the lung, so it's the pulmonary. And it's going away from the heart, so it's an artery. This is the one we need to be a bit careful about because it's an artery, it's going away from the heart. Then comes back in from the pulmonary vein. This is actually oxygenated blood. It's come from the lungs into the left atrium, down through the atrioventricular valves into the left ventricle, contracts with maximal force, and pushes blood up and out through. X is the... I've been ignoring you. Give me what X is. You've all gone to sleep. Semi-lunar valves. Spot on, Limity. You're the first, so... Semi-lunar. Half moon. And it comes out of here through the aorta. Okay, so... They like doing questions like this on the valves. The pressure changes here and where. What does this diagram suggest about the pressure in the atria compared to the ventricles at this stage of the cardiac cycle? Explain, give reasons for your answer. The atrioventricular valves are open, which means that the pressure must be greater in the atria than it is in the ventricles. So, because the valve is open, that is the explanation. So higher in the atria, lower in the ventricles, atrioventricular valves are open. 2 marks. Same question, different format. This is the volume of blood in the right ventricle, so over here in the right ventricle, and we've got time and we've got the volume. It doesn't display very well, but you can work with me. At 0.2 seconds, what are the valves doing? So at 0.2, we're going from 148 volume, 103 down to 70. So the volume is decreasing in the ventricle. So if the volume of the ventricle is decreasing, it must be emptying. So the blood must be coming up here, which means the semilunar valves are going to be open and the atrioventricular valves are going to be closed. At 0.6 seconds, what's happening? The volume of the ventricle increases, 5598125. So the volume of the ventricle is increasing. Blood must be filling the ventricle from the atria. So the atrioventricular valves must be open, and therefore this is just pump blood out. There'll be blood sitting in here that gravity will want to bring it down, so the pressure here will be greater than beneath. So these will be closed. Arteries and arterioles versus veins and venules. Lumen, this is often confused. This actually has a narrower lumen, and this is a wider lumen. Yeah. Muscle, we can say more, less, almost no muscle in the veins or venules. And treat arteries and arterioles as basically being exactly the same. Same as you treat veins and venules. Venules just means small, arterioles just means small. What are sphincters? I'm coming to that, Imogen. I threw this in just as a bit of a curveball, but it's I'll explain in just a second. Valves, none or not present. Present. Sphincters are little rings of muscle. Your asshole is a sphincter. So it's basically anywhere you've got a ring of muscle that's controlling the diameter of. This word sphincter isn't massively important. A lot of you are freaking out about that. But you need to know about vasodilation, vasoconstriction, so that if you eat a meal, you haven't got enough blood to be everywhere all at once. So after you eat a meal, you divert more of your blood to your digestive system so you can process and digest it. If you're going to go for a run, your muscles need more of that blood. You don't have enough blood to be everywhere at once. So these are like the little gates, the switches that say, my muscles need blood, then I'm going to divert blood to the oxygenated blood to this area. And I'm going to shut off blood supply or reduce blood supply to somewhere else. So the sphincters are the gateways that do this. I threw this in there because I saw it on a past paper recently. So we could say these are present and these are absent. So these control where blood is going to go, where the oxygenated blood is going to go. I don't know which past paper more, nor I've looked at probably 50 this week. Elasticity, again, we can say more elastic, more recoil, more elastic fibers, less elastic or not very elastic. Thickness of walls, thick, thin. I think that's it. Maybe we could draw a diagram. So with an artery, we've got a folded endothelium, endo inside lining. And then we've got some thick muscle around the outside. And whereas a vein, we're going to have a wider lumen and a thinner wall and almost no muscle. So with an artery, we've got this folded epithelium, because if we think, like when the heart pumps, when you get ventricular systole and the heart pumps, that's like contracting, it's going to have high pressure in your arteries. It's going to, like in a water balloon. If you fill a water balloon, it's going to stretch out in diameter like this way, and then when the heart stops pumping, you turn the tap off again, it's going to recoil. That elasticity is going to help squeeze the blood along that tube, because it can't go backwards, it can basically only go forwards because there's loads more blood behind it, and that's going to help even out the flow. It's going to help keep a consistent blood flow. What sort of questions to ask about arteries and veins? Basically, like the other tables, they can ask comparison questions like, what do you think? I wonder if I put one in here. I might have done. I think more or less. Did I go through all my pins? Biodiversity. Okay, so we can do this. Last one. I didn't do anything on biodiversity. Okay, so first of all, my comment here. This is a 3-mark question. You've got so many lines. Do not be put off by how much space they give you. 3 marks is 3 sentences. You could potentially get 3 marks in that much space. I would try and do that myself on a past paper. Absolutely, for sure, I would. Like, do not be put off by that. You've seen the mark schemes. Look, in fact, you can see over here, I'm not going to move, so that's not, there are 3 points on the mark scheme, if you can see that right away over here. Like, that is it. And they are their space, their answer would take up 3 lines and 3 lines only. So do not get sucked in by the amount of space. You don't have to fill it. Put in as many key terms as you can and think about the order of your sentences, but it's not obligatory to write that much at all. On intensive farms, the farmers had removed hedges to increase land for grazing. This resulted in a decrease in diversity of birds on these farms. Give reasons why the removal of hedges caused a decrease in the diversity of birds. Okay, so we've got less habitat. When I'm doing past papers, I try and do hundreds of past papers. I try and do as many as I can, and my answers normally look like this. Decrease habitat, decrease niches. Decrease food available. We've got decrease nesting sites, maybe. Give reasons why hedges, removal of hedges, decrease food, so they need to eat. There's not going to be an increase in predators. What causes it? A lot of this is like common sense. What causes numbers of the species to go down? Less food, less place, less children, more predators. Like, they are the kind of three major factors. So that's in past papers. If you're ripping through as many past papers as you can, as long as you know you can write a sentence that says there's a decrease in habitat, then you can be quick with yourself, and it makes past papers a lot less painful. Removal of species, types of plant or insect, fewer food sources, fewer habitats or niches. Again, 3 sentences. I think, ladies and gentlemen, boys and girls, that is basically everything in a snapshot. I don't think I forgot anything, did I? No, we did all that. We did all that. We did all that. We did all that. Oh, wow. Okay, so I overran by kind of 20 minutes, which was not my plan, but okay, sweet. So there will be a recording. I know a lot of that was like mega fast. What were the sticking points? We had DNA synthesis. Infection. Alex, I can't do infection because ZXL don't do it. I was going to go through that. Obviously, all the videos are in the course. If you want to rip through infection, exam board specific for you, then you can. I can't. I had to pick all the stuff that's basic. This is more, not everything. There is more stuff that's in common, but you can see how similar, like, that's all going to make sense to you. Then it's how similar the exam boards are. It seems so little when you look at it like that. Well, I haven't obviously covered every single thing that can be on the exams tomorrow, but I'd say it's a lot of those words are going to be in your paper 1 for sure. Okay, give me a usefulness score out of 10, and don't necessarily be overly polite, but like was that useful? 11, 8, 9. Okay, so like lots of 8s and 9s, and a few 10s. Okay, so like again, when we will do some questions, I'm not going to run away just yet. We've got a few minutes. I do have an appointment not long after 7:30, so I can't hang around forever. Do let me know in the comments, how can I make this better? I'm here to help you guys out. So the more details you give me and what you like and what you don't like, the more I can tailor all the other webinars. I've sketched them out, I've planned them, I've done the initial plan, but I can tweak them. I like responding to your feedback because you are the people that want to learn from this. Do I have any set to AQA biology papers? Zoya, I will be getting them. Actually, I don't have them on my hard drive yet, but I'm being sent them by an AQA examiner. More question applications. Love the questions. Predictive. I'm not going to do any predictive questions. People have been asking me for predictions on the papers. Like, the new spec, there's so few things going out there. Know your core topics. Know your biological molecules. Know your transcription and your translation and your DNA synthesis. And you know how mutations can affect proteins, because these are, like, there are bits of the course which kind of out on their own and they don't link up too much. All the stuff that links in together, you will get asked at some point. It will come up in some context in a question. 25 mark Zoya, I'm doing a full presentation, a full hour and a half on the 25 mark essay. When will be an AQA specific? All the schedules are on that page. I'm not going to answer scheduling questions because they're all available. You can go check them out for yourself. You're all doing A-level. You're smart enough to do that. How much AS content will be in the A2 exam? I'm doing a webinar on exam technique on Saturday. If it's not Saturday, trust the schedule that's online, because I can't remember off the top of my head. I think it's Saturday. In the beginning of that, I'm going to go through a little bit of what the different exam boards, what you need to know for the exam board. Have a copy of your specification downloaded. If you don't have a copy of the specification that you're doing, download one, so you've got a digital copy, and 2, print one out. Just not all of it. It's like a 90-page document. The pages that tell you what you need to know. So your Bible. chromatid versus chromosome. Oh, and DNA replication. Maybe I'll go over that. Okay, sense and antisense and DNA. Okay, let's maybe that was the most commonly, most common. Where in fact, let me put it next to where it belongs. Over here. Okay, so the terminology around chromatids and chromosomes is pretty horrible. This is a chromosome. Normally, if we could see a chromosome, this is what a chromosome would look like. We can't normally see a chromosome because during interphase, they haven't condensed yet. And so if this is our nucleus, DNA just you can't see it. It's just a mesh of stuff. If chromosome were to condense, it would look like this. And lots of people get massively twisted up over chromosomes and mitosis and whatnot because they think that chromosomes look like this all the time. Chromosomes only look like this X shape when they're replicated. So this half, these two are mirror images of one another. They look like this after the DNA is replicated. So this, the terminology is horrible. This is a chromatid. We can, when it splits apart, it's a chromosome on its own. This whole thing, when there's two of them, the literature says is also a chromosome. So they all basically have the kind of the same names. I personally call this a double chromosome. That is not a key term. But a double chromosome tells us that it's got both wings, it's got both halves there. And one half of a double chromosome is called a chromatid. As soon as the centromere breaks and these split up, and we have this here and this here, this is now a chromosome. And this is a chromosome. So there's lots of overlap between these key terms. Sister chromatids just means that this one and this one are, they contain exactly the same genes and exactly the same allele. So this, whatever this allele is here, let's say this is little b for blue eyes, this one will also be little b for blue eyes. If we have another, the homologous pair of this chromosome, which I've just drawn terribly, we go to the same locus down here. It's so badly drawn, I'm going to redraw it because that is terrible. It needs to look half the same. Okay, so I should have done it in blue. Okay, so at the same locus on its homologous pair. So let's say this one came from your mother, and this one came from your father. It contains the same gene. So this gene is also the eye color gene, but this came from another person. This came from, let's say, your dad, and this one came from your mum. So this could be the brown eye allele. Eye color is not monogenic, but I'm using it as an example. So a homologous chromosome. So this is, let's say, chromosome number 12. And this is chromosome number 12. That means they contain the same genes. So they contain the same genes. So they've got the eye color gene. It might have the curly hair gene or whatever it might be. So they've got the same genes, but the alleles don't have to be the same. They obviously can be. They can be the same, but they're not necessarily the same. DNA replication. Diploid and haploid. Let me do the DNA. Let's do one question at a time. Okay, so geez, how am I going to draw this? Okay, so we've got our DNA. There's a helix up here, or terribly drawn. What enzyme unzips the DNA? This is DNA helicase. Okay, and DNA polymerase. This is going to be the sense strand or the template strand. Then it runs in. Okay, so the active site of the enzyme. So we're going to get our DNA nucleotides pairing up. Let's say we've got C, T, G, A. We're going to get our nucleotides pairing up here. We're going to have G, A, C, T. And these nucleotides are going to be joined to form phosphodiester bonds, and that is going to be done by the enzyme. It's going to be sitting here, doing this one, fusing this one here, forming phosphodiester bonds, and this enzyme is DNA polymerase. The polymer making the polymer, A's enzyme making the polymer of DNA, DNA polymerase, forming the phosphodiester bonds. So it's like joining up this bit here. Now, this enzyme has an active site. That means in this strand, our pentagons in our nucleotides are pointing up. So we can think, so this is directional, and therefore the pentagons on the nucleotides in these guys are pointing down. So these are anti-parallel. So the active site of DNA polymerase is specific. It's a tertiary structure. It's specific like all other proteins and enzymes are, which means it can run continuously in this direction. It can just like take the block from over here and catalyze the reaction. Take the nucleotide, catalyze the reaction. It's built, its active site is like feeding it through, and it can do it in this direction. Okay, so if this strand, the template strand or the sense strand, is over here, then this is the antisense strand, and this one is running in this direction. Now this means the enzyme, so the, okay, so let's go, I'm going to not do its pairs because I'm going to got more space down here, so I'm just going to make up some more bases. C, C, G, T. Okay, this time it's not running, this was running from top to bottom, yeah, because the active site can do it. The active site is now going in the, would be going backwards, and it can't go backwards. It's like a mirror image. It's not the same that way around. So this has got to go from in this direction. So this is going to do A first, and then C, and then G, and then G, and we're going to make a little sequence of DNA, a little patch of DNA. And what's going to have to happen is we're going to form little patches, and then it's going to have to break off and then do another little patch, and then do another little patch, because it can't go backwards, basically. So the direction of the active site is directional. And DNA ligase, who was ever asking me about DNA ligase, you build a little chunk going in the right direction, and it breaks off, and you build a little chunk going in the right direction. And DNA ligase is the enzyme that joins up the chunks. Any questions? That was not very well. I mean, that is the major points on DNA replication. Remember, I went through already. DNA helicase unzips and breaks the hydrogen bonds. Free nucleotides are added. DNA polymerase joins the nucleotides to form phosphodiester bonds. From both strands are copied. Okay, loads of questions. From last year's HOA paper, the mark scheme of a tick box says that bacteria have enzyme molecules. What are enzymes molecules? They're just enzymes. They are a molecule that is an enzyme. Bacteria have enzymes too. They can make proteins, they can make enzymes, they need them for working. So there's just emulsion test in Edexcel. Let's have a very quick goosey gander in here. Biochemical tests, they're often called. No, I don't think it is. I searched for emulsion already. Okay, so it's bound to be in here, if it's in here. Are any of the tests in here? I don't think they are. My head is exploding. In the new spec, 4 different exam boards, my head is trying to... Sedan, I'm sure Sedan 3 is not going to be in there. It's like never mentioned by anyone. No. The AS spec, the content, the indicative spec in the content for the... This is the AS Edexcel spec. You guys are awesome helping me out. Emulsion, no. Okay, in which case, my okay, so something that is quite useful. This is going to be in the exam technique thing. This is obviously the one I was looking at earlier. They Edexcel publish what they expect their command words to be. So if you're not sure, I'm going to cover all that in the exam technique webinar at the weekend. Have I done a 2 cheat sheet? I'm still writing it. I'm still building it. I might put out like a draft version for you guys because I might put out a draft version, but I want it to kind of look nice. I don't want it just to go out with my name on it and it look like a dog's breakfast. But the webinar is recorded, Zena. It will be. What is the cheat sheet? It's currently downloadable from our homepage, tutors.co.uk. I think down the bottom you can download the cheat sheet. Go and do it. It's awesome. Did I get to look at the Edexcel article? Not yet, Beth. I've still got 88 emails in my inbox, which I haven't yet finished. I know you don't care what it looks like, but it will get distributed, and I don't like things looking nasty with my company on it. Isabel, it's basically it's the stuff kind of similar to what we did today, but like in PDF form. So it will go through the classic DNA replication, and it will give you very, very short bullet points that I put together, basically like the perfect answer on the classic generic questions. There's some of the immunology on there, Alex. So if you wanted the immunology stuff, phagocytosis, activate T cells, B cells, all that kind of stuff, then that's on there. Where can you find the AQA spreadsheet? Hassan, can you tell me what you mean by the spreadsheet? I don't quite know what you mean by the AQA AS spreadsheet. Does AQA paper 3 have all 8 topics? Correct. AQA paper 3 does have all 8 topics. I'm doing a webinar on the AQA paper 3 though, so don't stress too much. Any more exam technique videos like the one on proteins, like summaries? There are a few in the course. I'm not going to be able to make any more. I'm going to spend my time doing live webinars and stuff like that because I can help you out a bit more. Oh, the cheat sheet. Oh, not spreadsheet. Okay. What was your question about the spreadsheet? It's already gone off the page. Where can I find it? It's not exam board specific. Feedback from you guys is that you want me to make it exam board specific. I will do that next year. It takes me a long time to write those things. Obviously, there's lots of information on there. The cheat sheet is on the homepage, tutors.co.uk. I think it's down the bottom is the button. Do I know why AQA doesn't have a maths and stats skills booklet? No, because they're annoying. I want them to make one, but they don't have one. But the maths and stats skills are set by Ofqual, who basically regulate everyone, so they're more or less the same for all of you. So you can, if you're an AQA student, you can use the OCR and Edexcel ones. They're pretty good. Is there a 25-mark essay video? I did make one. It was terribly out of focus, and I don't know if I uploaded it or not. I'll check. If it's there, it'll be in the AQA A-level course. AQA Edexcel 9-marker tips, so your extended question in Edexcel is 9 marks. Yeah, think about it just a little bit. Break down the question. Go do the exam technique webinar with me on the weekend because basically lots of key terms. Think about the order, think about the sequence, and loads, at least 1 key term per sentence, basically, and you want at least 9 key terms, therefore, in your answer. Can I upload it? I don't think you've put it up. I don't. Are you talking about this cheat sheet? I don't know what you're talking about, Aruba. Daisy, would you recommend AQA past papers even though the spec is... Yes, the old AQA past papers. For all of you, the spec is, let's say, 75% the same, 25% different. The modules are rearranged and they're not everything's in the same place. But the old past, because there's so few past papers now, the old past papers are a massive resource. You will need to be confident with your specification. So download your specification. If you're getting questions that are on things that you've never seen before, then you need to know that that's from the old specification and not on the new specification. But absolutely, the old papers are going to be very, very valuable. I can, like, a lot of the stuff that I pull, I know what's on the new and what's on the old, so it's a bit easier for me, but I pull most of my paper questions from the old spec just because they haven't changed very much. I wonder if I can show you. Yeah, if you Google, where is it? This is for AQA students only, I'm afraid. Uh, Summary of changes. This is the URL. You can take it off the recording. This tells you what's different between the AQA old spec and the AQA new spec. Over half of you are AQA students, so I don't feel too bad for just a second. EdXL and OCR don't have one of these, which is rather annoying. So you can look up. So carbohydrates is now, there's no new content. So that tells me that there's no new content been added. All the content from what's gone, you don't have to know what about lactose intolerance anymore. What's changed, and it tells you what's changed. What's the same, tells you what's the same. Proteins, what's the same. Okay, so it tells you what's the same, what's different. So that's useful if you're using the old past papers. OCR don't do one, I'm afraid. Yes, I would use past papers like right now, the last month before your exams, or last couple of days before your exams if you're an AS student. Like past papers will be more and more and more forming the content of your revision. If you get questions wrong, that's okay. Go back and mark your past papers carefully. Change color, add in all of the questions, all of the answers, sorry, that you didn't get or you got wrong. Then I would spend as much time looking at the answer. Why did I get it wrong? Did I not know? I just didn't know the content. Did I misinterpret the question? Did I go off topic? Did I know the answer? Was the answer in my head, but I didn't write it down? Did I not include the key terms? Analyze. I spend almost as much time marking my past papers for practice, and it's going to show you what you know and what you don't know. Don't spend the time. People love revising what they know because they do the test and they get, yeah, self-gratification, I know this, and they avoid doing the stuff they don't know, which if you look at it from an outside perspective, stupid. You should be working on the things that you're finding the hardest. Do you think that will be similar for the EdXL spec? What's changed since they are told the same people that they need to include it? I don't quite understand your question, Beth. Sorry. Um, should you do past paper questions by topic or whole past papers at a time? As we get closer to exams, I would do whole past papers. I mean, if you can find question packs, there are some out there. I'm putting some together. I'm hope they're with my graphic designers to try and make them look a bit nicer. I've put together some of the biological molecules and stuff like that. This time I'd get used to your exam timings and your practice. I'd sit down and try and do a past paper, a complete past paper. If you find that you've come to a topic and you've just not revised it, have a quick go. Don't beat yourself. Don't know it because you haven't revised it yet. But just what can you remember based on the key terms? Put a few bullet points in. Trying to remember it, and that testing effect it's called, is very, very powerful. When do I think the A2 cheat sheet will be available? I've got lots of planning to do. Okay, I'll try and get a draft done by the end of this week. I'll try and get the draft with the graphic designers for the weekend. How's that for a tentative promise? Will the summary of changes from AQA be similar to EdXL? No, I wouldn't say it's any use to you really at all because it's the specific spec changes. In principle, yes, but the actual fine details, no. Not enough to rely on anyway. Do I recommend doing past papers before you revise topics? Yes, I would. Because you'll be amazed sometimes you'll actually remember it quite well and then you'll give yourself confidence. Sometimes you might not remember it. That's okay too. If you've not revised it, there's huge amounts of content in biology, but testing yourself before you get there, when you mark the question, just bullet point what you think you can remember. When you mark it, put in those notes. That is quality revision. Again, when you maybe mark the question and it's on a topic you've not done much study on, read the question again. Maybe you got 0 out of 6, 0 out of 9. Don't stress. Read the mark scheme. Refresh yourself of that. Refresh the memory to yourself instead of just copying it. Again, try and learn the mark scheme. Spend 5 minutes learning and doodling some notes. Flip the mark scheme upside down and then re-answer the question. It's more work. It's more effort, but that is the way. You've got to learn those mark schemes. That's essential. Yes, definitely do as many past papers as you can, basically. So I'd revise a topic, maybe do some questions on the topic, and then maybe do at least one past paper a week minimum, I think, between now and exams, more if you can. And you might have to do some of the old past paper questions to do that. I don't normally recommend doing past papers from other exam boards because there's normally loads of past papers available. Now, there's not many past papers available. The new style of exam is the same for OCR, EdXL, WJEC. Some of the content is different. EdXL don't do immunology, for example. So if you're an EdXL student doing an OCR paper, you'll be like, phagocytosis, don't know about this. But that's where you need to know your spec and be confident with your spec. So then you could use the other past papers from other exam boards to get more practice, because exam practice, the biology A-level exam, this is something I'm going to mention at the beginning of the next webinar, tests you how good you are at the biology A-level exam. It doesn't test you how good a biologist you are. So you have to be good at doing the exam. That is what you are being tested on, not how good you are as a biologist. So I've massively ranted on. I hope that's been helpful. I've got to be online to speak to someone else in Canada in 3 minutes time, so I can't stay any longer. Thanks, guys. Again, leave as much feedback in that comment section. I don't really care. Well, you can fill in the rest of it, but I want those comments, feedback about this. I'm going to be doing one like this for A2. Let me know what you want so that I can tweak it and change it and make it as effective as possible, so I get as much content in to help you get as good a grades as you possibly can. So good luck. If you've got the exam on the AS exam on Thursday, is it? Awesome luck. Be confident, do past papers, work hard, but yeah, set yourself blocks. Don't try and work all the time. Work for an hour, set yourself a target, do that bit, get up, take a break. You can't work forever.